Hello everyone, this is Sarvani Sistla, third year postgraduate student in the Department of Pediatric and Preventive Dentistry from Paniniya Institute of Dental Sciences. Today I am here to discuss the process of physiological root resorption. It is one of the most common terms we concurrently use at a drop of a hat while conversing about primary teeth. But the main question here is, to what extent do we understand the actual process? My motive today here is to throw some light on this process in depth. Process can be understood under few headings, which include the introduction, the key cells involved in the process, the actual process that occurs in the primary teeth which present with the successor and which present without one. Lastly, few related articles and summary. Coming to the introduction, the term physiology means the branch that deals with the functions and activities of life. In short, these are those processes which occur naturally within the body. On the other hand, resorption means destruction or disappearance. Generally, the mineralized structures of the teeth are protected against resorption by the predentine and the odontoblasts within the pulp cavity and the pre-cementum and the cementoblasts on the root surface. Both these layers form a barrier that prevents resorption and osteoclasts from adhering or resorbing the unmineralized matrix. Because of the inhibitory effects of the organic pre-cementum and predentine, even in the presence of inflammation, an intact root can be appreciated which is resistant to resorption. In situations where these protected structures are damaged or removed either physiologically or pathologically, multinucleated giant cells, that is the osteoclasts, colonize on the root surface and cause resorption. Key cells, which include osteoclasts, odontoclasts, monocytes, and macrophages. These cells are of crucial importance for the process of resorption to occur. Osteoclast is a Greek term where osteos means bone and clast means to break. It is basically a type of cell that breaks down the bone tissue. They are motile and multinucleated with nuclei ranging from 2 to 12 per cell. They are derived from the hematopoietic cells of monocyte macrophage lineage with a lifespan of 2 weeks. They recruited to the site of injury with the release of pro-inflammatory cytokines. To perform their function, osteoclasts must attach themselves to the bone surface. They have a ruffled border which helps them to bind to the matrix adhesion proteins and produce the resorption pits called the Hauschilt's lacunae. They can be differentiated histologically by the immunohistochemical stains for the tartarate-resistant acid phosphatase. Coming to osteoclast formation, numerous cytokines have been shown to regulate osteoclast formation and function. Out of all of them, these are the 5 important cytokines. The first 3, that is the OPG, RANKL and RANK, will be dealt in detail in the later part of the presentation. But for now, RANKL and RANK are 2 important cytokines which positively regulate the osteoclastogenesis, leading to the increased expression of new osteoclasts, eventually increasing the resorption rate. While on contrary, OPG negatively affects this process, leading to decreased expression of new osteoclasts. MCSF is a macrophage colony stimulating factor, while CFMS is its receptor. Coming to the process, once the osteoclast precursor cell is formed from the hematopoietic lineage cells, they express colony stimulating factor receptors as well as RANK. This CFMS and RANK bind to the stromal or the osteoblastic cells, eventually leading to the expression of RANKL, OPG and macrophage colony stimulating factor. This process is mediated by a few factors such as parathyroid hormone, vitamin D, interleukin 1, 611 and tumor necrosis factor. At this stage, if the expression of RANKL is greater than OPG, the osteoclasts are formed, leading to the increased bone resorption. While in contrary, if the stromal cells produce OPG in greater amounts when compared to that of the RANKL, the OPG binds to the available RANKL and the new osteoclast formation is prevented. Also, in cases of inflammation, there is increased activation of T lymphocytes which produce both membrane bound and free RANKL, leading to the osteoclast mediated bone resorption. Also, in cases of inflammation, when there is increased expression of interleukin 1 and TNF alpha, these 2 factors directly stimulate the osteoclast precursor cells to get converted into osteoclast, increasing the rate of resorption. Coming to the functional elements of fully differentiated osteoclast, generally the osteoclasts attach to the bone via podosomes which contain V3 integrin. Podosomes are motile organelles of few cells such as osteoclasts and certain immune cells, while integrin is a transmembrane protein which is involved in the adhesion of cells. Basically, the complex of podosomes allows the osteoclast to attach itself to the bone. Inside the cell, the H plus ions are produced by the carbonic anhydrase 2 enzyme. These H plus ions are transported to the resorption site via the H plus ATPase pump. This H plus ATPase pump, coupled with the chloride channel, maintains the transmembrane neutrality. Also, there is excessive production of bicarbonate by the carbonic anhydrase 2 enzyme. This bicarbonate is exchanged passively for chloride, which is the chloride and the bicarbonate exchanger here. The chloride ions as well as the H plus ions present inside the cell are transported to the resorption site where they create an acidic environment. In this acidic environment, the enzyme cathepsin K is activated, which is vital for the removal of the organic components of the bone, leading to the resorption. Various cytokines are involved in the formation of osteoclasts, but here we will be discussing three important cytokines, out of which the first one is RANKL. It is a receptor activator of nuclear factor kappa B. It consists of 616 amino acid peptides. It is a member of the TNF receptor superfamily. Its potent stimulators are the macrophage colony stimulating factor, transforming growth factor beta, vitamin D3 and dexamethasone. Its inhibitors are interleukin 4 and interleukin 6. Is the RANKL. It is a receptor activator of the nuclear factor kappa B ligand. It is again a member of the TNF receptor superfamily. It is produced by the marrow stromal as well as the osteoblastic cells. It is selectively expressed in the actively resorbing primary teeth periodontal ligament, but not seen in the permanent counterparts. Its potent activity is that it is a stimulator in the formation of osteoclasts from the precursor cells. Also, the bone resorbing activity of mature osteoclasts is altered by them. The osteoprotectin is again a member of the TNF receptor superfamily. It consists of 380 amino acids. It is produced by the marrow stromal as well as the osteoblastic cells. Its activity is that it's a potent inhibitor of the formation of osteoclasts. That is, it acts as a decoy for the RANKL. It binds to the available RANKL and inhibits its function of forming new osteoclasts. Few factors such as TNF alpha, interleukin 1, TNF beta, bone morphogenic protein and few steroid hormones upregulate the expression of OPG, while glucocorticoids, immunosuppressant drugs, parathyroid hormone, prostaglandin E2 suppress the expression of OPG. The important point here to note is when there is lack of OPG, osteoporosis is seen and there is increased number of osteoclasts, while overexpression of OPG results in osteopetrosis and decreased number of osteoclasts. Coming to the resorptive process, which is mediated by the osteoclasts, it can be easily termed as a bimodal process. That is, initially there is dissolution of the inorganic crystal structure followed by the degradation of the organic collagen, principally type 1. Coming to the dissolution of the inorganic crystal structure, when the pH is below 5 at the resorptive site, there is rapid dissolution of the hydroxyapatite crystals. A polarized proton pump along the ruffle border is formed, and the enzyme carbonic anhydrase 2 plays an important role here. As already discussed in the functional elements of the osteoclasts, inside the osteoclast, the carbonic anhydrase 2 enzyme forms H plus ions, and these H plus ions are transported to the resorption site through the polarized proton pump. Coming to the degradation of the organic structure, three groups of proteases are involved here, which include the collagenases, the matrix metalloproteinases and as well as the cysteine proteases. The collagenases and the matrix metalloproteinases act at a neutral pH, while the cysteine proteases act at the acidic pH. As discussed, cysteine proteases appear to work closer to the ruffle border because it is acidic here, that is at the resorptive area, while collagenases appear to be active at the resorpting site because the pH is closer to neutral here, because of the buffering capacity of the dissolving bone salts. And to summarize the whole process, once there is proliferation and differentiation of mononuclear precursor cells from the blood, there is production of osteoclast precursor cells. These cells fuse, eventually forming the osteoclast. This is the formation of osteoclast. Coming to its function, its function is mediated by RANKL and OPG. If there is more production of RANKL, there is osteoclast activation, while if there is more production of OPG, there is no osteoclast activation and there is suppression. Coming to its attachment to the bone, the osteoclast attaches itself to the bone by the podosomes, forming a sealing zone. Within the cell, by the action of carbonic anhydrase 2, there is production of H+, which are transported to the resorptive site, forming the acidic medium here. Once they are transported to the resorptive site, there is degradation of the organic matrix as well as the destruction of the inorganic crystal structure, eventually leading to resorption. Now, let's learn about the second important key cell, which is the odontoclast. Odontos means tooth, while clast means to destruct. These are the cells which destruct the mineralized structures of the tooth, eventually leading to their resorption. Odontoclasts are thought to differentiate from the circulating progenitor cells. Such progenitor cells reside in the dental pulp and periodontal ligament. They share many similar characteristics with that of the osteoclasts in the expression of cathepsin K, cathepsin D, tartarate resistant acid phosphatase, H plus ATPase pump, and formation of a clear zone and ruffled border. In the physiological primary tooth resorption, mature odontoclast formation only occurs after the attachment to the resorption site. The cells from the pulp, pedial, cementum and dental follicle mediate the formation of osteoclast, odontoclast and eventually the root resorption. They are involved in the orchestration of the essential molecule in the OPG, RANK and RANKL system. Apart from this, pedial cells also mediate the systemic regulation of osteoclast formation through parathyroid hormone and also by increasing the expression of RANKL. All these factors eventually induce notch signaling, promoting the odontoclastogenesis. Now, let's understand the process of root resorption caused by odontoclasts. Once there is a stimulus, either physiological or pathological, there is a local increase of cytokines as well as the odontoblasts and fibroblasts are activated. With the local increase of cytokines, the T cells get activated, eventually expressing RANKL. These lead to the differentiation and activation of the pre-odontoclasts, eventually converting into odontoclasts. With the activation of odontoblasts and fibroblasts, there is expression of RANKL and interaction with the mononuclear progenitor cells. They lead to vasoactive, chemotactic and cellular effects with the recruitment of active odontoclasts. Both these processes result in root resorption. Of cells which play a crucial role in the resorptive process include the monocytes and macrophages. They are in similar structure to the osteoclasts. They can also become multinucleated giant cells, but they lack a ruffled border and do not create any lacunae. Once the monocytes are recruited to the site of injury by the release of pro-inflammatory cytokines, there the monocytes get differentiated into macrophages. These macrophages get migrated and recruited by the action of the macrophagic chemotactic factor, which is derived from the bone and the tissue breakdown products. We all are well aware that physiological root resorption is one of the basic features of primary teeth which is not exhibited by the permanent teeth. But how many of us know the exact reason for this to happen? Now let's discuss a few reasons for this to occur. The first one could be the extracellular matrix protein distribution pattern, which is different in primary and permanent. Lee et al. In 2004 stated that osteoclast and odontoclast activation may be extracellular dependent. The second possible reason could be the expression of the RANKL, which is seen more in the periodontal ligament cells of the actively resorbing primary teeth and not by the permanent teeth. The third possible reason could be the colony stimulating factor, which are significantly expressed in the primary dental pulps and not by the pulp tissues of the permanent teeth. Coming to the fourth possible reason, it could be because of the RANKL expression, which is also high in the pulps of the fibroblasts, odontoblasts, cementoblasts and aminoblasts of the primary teeth and not by the permanent teeth. The consistency and symmetry between the right and left sides of the mouth regarding the exfoliation timing of the primary teeth and the emergence of their permanent successors are indeed fascinating. This suggests that the shedding of these primary teeth as well as the eruption of the permanent teeth are coupled events which could be programmed. The pressure from the erupting permanent tooth is believed to play a contributory role in setting off the resorption, but the presence of a permanent successor is not a prerequisite for this process to occur. The primary teeth without a successor do resorb eventually, although their exfoliation timing is a little later than usual. Now, let's understand the difference between the resorptive process in teeth which present with a permanent successor and those which present without one. Root resorption of primary teeth starts at the root that is closest to the permanent successor. For example, in case of anterior teeth, the completed crown is found lingual to the apical third of the primary predecessor. The eruptive force here is labial and incisal in direction, and thus it causes resorption of the lingual surface of the apical one-third of the primary tooth root. That is, here the lingual surface of the root is first resorbed. Once there is progression and resorption of the labial surface of the root, the permanent crown is found underneath the primary tooth root, and it then extends its resorption in incisal direction until the primary tooth exfoliates and there is eruption of the permanent tooth into the oral cavity. In some instances, permanent mandibular molars will not move forward, that is labially enough, and this causes the failure of resorption of primary teeth, eventually leading to retained primary teeth, and the permanent incisors are erupted lingually. In cases of primary molars, the developing permanent teeth are found lingually initially to the predecessors, and as the growth proceeds, the developing tooth moves under the divergent roots of the primary tooth root and causes resorption. The position and size of the follicle affects the pattern of root resorption. More than 36% of the primary mandibular second molars show uneven root resorption. This could be because of the highly curved and divergent roots, and also the interroot distance is often larger than the size of the follicle of the successor. Depending on the position of the follicle of the successor, unequal influences may be applied on the tooth roots. This can explain the reason for more than one-third teeth of the primary mandibular second molars showing uneven root resorption. This is even appreciated in the upper primary second molars, where the highly divergent palatal roots show more than 56% of uneven root resorption. This uneven root resorption is shown more by the primary second molars when compared to that of the primary first molars. This could be because of the smaller differences between the interroot distance and also the size of the crown of the permanent successor. Although never proven, it was believed and is still assumed that the pressure from the erupting permanent tooth causes differentiation and activation of the odontoclasts. Among the components of the erupting permanent tooth, the dental follicle and stellate reticulum seem to play important roles in the resorption of the deciduous roots. Since 1930s, Cronfield suggested that the dental follicle is responsible for the resorption of the roots of the primary teeth. Elegant studies conducted by Mark and Cahill stated that dental follicle of the permanent tooth is responsible for the root resorption rather than the permanent tooth itself. In their studies, the developing tooth crown was removed and inert substitutes of teeth such as silicone and metal replicas were placed in the dental follicles. The tooth erupted successfully, that is, the tooth substitutes erupted successfully into the mouth, indicating that the dental follicle regulates and coordinates the resorptive process. In cases where the dental follicle was removed, the teeth were prevented from eruption. It can be noted that the development of the eruption pathway, which involves resorption of the bone and resorption of the primary teeth, is a genetically programmed event, and it does not depend upon the pressure from the erupting tooth bud. The dental follicle cells and the bone stromal cells adjacent to the dental follicle express RANKL. The expression of RANKL is also upregulated by the expression of the parathyroid hormone related protein. It upregulates the expression of RANKL and downregulates the expression of OPG. This indicates that the dental follicle cells can replace the osteoblasts in the tooth microenvironment and coordinate the differentiation and activation of monocytes to osteoclasts and odontoclasts. Basically, it says that the dental follicle cells can express RANKL and eventually form osteoclasts and odontoclasts out of it. This happens in both the dental follicle cells as well as the PDL cells. It doesn't come as a surprise of this happening in the PDL cells because PDL is anyways a derivative of a dental follicle and it acts according to it. At a critical time of the eruption process, the cells of the stellate reticulum secrete parathyroid hormone related protein. It is a developmental regulatory molecule which is required for the tooth eruption. This binds in a paracrine function to the neighbor parathyroid hormone related protein receptors expressed by the cells in the dental follicle. Similarly, stellate reticulum also secretes interleukin 1A. This binds in a similar fashion to the adjacent receptors found on the dental follicle. Now, these stimulated dental follicle cells secrete a few receptors, that is the monocyte recruiting factors such as colony stimulating factor 1, monocyte chemotactic factor and vascular endothelial factor. Under the influence of these monocyte recruiting factors, monocytes arrange themselves adjacent to the rich vasculature, mostly in the coronal region. Once the environment is favorable in the dental follicle, monocytes fuse and subsequently differentiate into hard tissue resorbing cells, that is the osteoclasts or the odontoclasts, once as they come in contact with the RANKL expressing cells. This is about the action of dental follicle and stellate reticulum on the formation of odontoclasts. But recently, even cementoblasts have shown to express levels of RANKL and OPG, which is modulated by parathyroid hormone related protein. Interestingly, under non-resorbing conditions, cementoblasts seem to secrete a larger amounts of OPG. This could be one of the possible mechanisms by which the cementum is protected against the resorption when compared to that of the bone. Also, Sahara et al. Observed an intimate cell-to-cell relationship between the cementoblasts and the pre-odontoclasts. This can trigger a cytodifferentiation of the pre-odontoclasts into active and mature odontoclasts, which allows their attachment and fusion to the root surface. Coming to the pulp status during resorption. During the resorption of primary roots, the pulp remains in normal condition, at least initially. It does not seem to participate in the process of resorption. The odontoclasts are usually not found inside the pulp, or at least until the root resorption is close to completion and not before the resorption level has reached approximately 1 mm below the cemento-enamel junction. During this phase, chronic inflammatory cells, that is the T and B lymphocytes, infiltrate the coronal pulp and the odontoblasts begin to degenerate. Along with this, there is bacterial incursion through the gingivo-dentinal junction and accumulation of inflammatory cells into the pulp tissue. Following the degeneration of the odontoblasts, the odontoclast cells start resorbing the exposed dentine and both the dentine as well as the predentine from the inner surface. Initially, the resorption process starts at the pulpal floor, then towards the pulpal walls, and then it resorbs the pulpal horns. Also, the activated T lymphocytes, odontoblasts, and the pulp fibroblasts express RANKL. They interact with the RANKL receptor, eventually forming the active odontoclasts. These odontoclasts resorb and remove dentine and enamel, leaving the crown of the primary tooth thin and fragile like a shell, which breaks and eventually exfoliates into pieces. Finally, let's understand the fate of the gingival epithelium and the dentino-gingival junction. Once the root resorption advances due to inflammation, the dentino-gingival junction and the gingival epithelium migrate apically. They move under the crown of the resorbing tooth, protecting the developing tooth bud from the inflammation of the remaining pulp tissue of the primary tooth. Events described above are initiated and coordinated by dental follicle of the permanent tooth germ. However, even the roots of the primary teeth that do not have a permanent successor eventually resorb. How this happens and why it happens faster in some cases and much later in others is largely unknown. Ordinarily, the root, as earlier mentioned, is protected by a pedial layer against the resorption. The pedial is mainly composed of collagen fibers, fibroblasts, and cementoblasts. The degradation of the pedial precedes the root resorption, and specifically removal of collagen fibers of the pedial is considered a main step in the initiation process of The resorption. Interestingly, differences have been noted in the composition of the PDL cells of the primary and the permanent teeth, which may explain in part at least the increased susceptibility of primary teeth to resorption. The main differences include the PDL cells of the primary teeth produce more matrix metalloproteinases 9 when compared to that of their permanent counterparts. Also, these PDL cells respond more to pro-inflammatory cytokines such as interleukin 1 and TNF alpha, which eventually enhance the MMPs but not the tissue inhibitors of MMPs. Once these matrix metalloproteinases act on the PDL, they cause a collagen digestion, which is considered one of the main steps in the initiation process of resorption. Also, these matrix metalloproteinases are produced by osteoblasts, PDL cells, odontoclasts, and osteoclasts as well. Which can explain the resorption in such teeth is that as the face grows in size, muscles of mastication enlarge. Once they enlarge, the forces that are applied on the deciduous teeth become heavier than the primary tooth PDL can withstand. Eventually, they weaken the primary tooth PDL, causing necrosis. This induces localized production of cytokines which recruit macrophages and monocytes there. Under the influence of few factors, these get differentiated and activated into active odontoclasts. Once the odontoclasts are formed, they damage the PDL layer which protects the tooth, and the process of resorption eventually starts. Etiology for mechanical trauma could be abnormal occlusion which occurs during the mixed dentition phase. That is, a permanent teeth in one arch occluding with the primary tooth of the other arch. And one more etiology could be the composition of the extracellular matrix proteins. The two main proteins include the osteopontin and bone ciala protein. They are associated with the odontoclast adhesion and activation. And they were found to be heavily expressed in PDL cells that surrounds the primary teeth when compared to that of the permanent teeth. They promote selective binding of the odontoclasts, leading to the resorption of deciduous roots. There are no accurate predictors for the survival of primary teeth without a successor. In some cases, they can be well maintained into the adulthood, whereas in other cases, they may exfoliate much faster. If a primary molar remains in the dental arch without no significant changes for 20 years of age, it is considered favorable prognosis for long-term survival. Here is the article which was published in the European Journal of Oral Science. There an experimental study was conducted in beagle dog to see the resorption rate in the primary molars without a successor. The main aim was to investigate the mechanisms of the root resorption in primary molars without successors. The results showed that the resorption in such teeth began much later when compared to that of the physiological resorption. Also, the pulp showed odontoclasts and immune cells nearing to the resorption site. There were few immune cells which were noted in the periodontal ligament area. They concluded saying that the dental pulp of primary molars as well as the immune cells may play a role in the resorption of such teeth. To summarize the whole process, the first important point to remember is that it is a genetically determined programmed event. The second point to remember is the key cells involved in this process are the osteoclasts, odontoclasts, monocytes as well as the macrophages. The third take-home point is that the whole process is an interplay between the RANKL and OPG system, where the RANKL increases the resorption rate while OPG suppresses it. The fourth point is that in case of primary teeth with a permanent successor, the structures which play an important role are the dental follicles, stellate reticulum and the cementoblasts. While in primary teeth without a permanent successor, the PDL and pulp cells of the primary teeth as well as the extracellular matrix proteins play an important role. The final and the most important point to remember is that the pressure from the erupting permanent tooth just has a contributory role and it is not a prerequisite for the resorption process to occur. I hope my effort helps you to dispel the myths you have about this basic process. Thank you.